Here are 5 USMLE style questions focused on mutations in DNA, including silent, missense, nonsense, and frameshift mutations, as well as point mutations and types of substitutions. 1. Which of the following mutations is most likely to result in a non-functional protein due to premature termination of translation? A. Silent mutation, B. Missense mutation, C. Nonsense mutation, D. Transition mutation, E. Frameshift mutation. Answer C. Nonsense mutation. Explanation. A nonsense mutation introduces a premature stop codon, e.g., UGA, UAA, or UAG, leading to the truncation of the protein and usually resulting in a non-functional protein. Missense mutations change a single amino acid but typically do not cause truncation. Silent mutations do not change the amino acid sequence, and frameshift mutations alter the entire amino acid sequence downstream of the mutation. 2. In which of the following mutations does the new amino acid share a similar chemical structure to the original amino acid, making it less likely to affect the protein function? A. Missense mutation, conservative. B. Missense mutation, non-conservative. C. Nonsense mutation. D. Silent mutation. E. Frameshift mutation. Answer: missense mutation, conservative. Explanation. In a conservative missense mutation, the substituted amino acid has a similar chemical structure to the original amino acid, making it less likely to affect the protein function. Non-conservative missense mutations, on the other hand, replace the amino acid with one that has a very different chemical structure, which is more likely to affect the protein's function. Nonsense mutations result in a truncated protein, and silent mutations do not change the amino acid sequence. 3. Which of the following best describes the genetic change in sickle cell disease, which is a result of a missense mutation? A. A transition mutation that results in a change from adenine to guanine. B. A missense mutation that results in the substitution of glutamic acid with valine. C. A nonsense mutation that creates an early stop codon. D. A frameshift mutation that results in a non-functional hemoglobin protein. E. A silent mutation that causes no change in the amino acid sequence. B. A missense mutation that results in the substitution of glutamic acid with valine. Explanation. In sickle cell disease, a missense mutation occurs in the hemoglobin gene, where glutamic acid is substituted by valine at position 6 of the beta-globin chain. This mutation alters the structure of hemoglobin, causing it to aggregate and form sickle-shaped cells, which is characteristic of the disease. This is a classic example of a missense mutation. 4. A mutation in the third position of a codon results in no change to the amino acid sequence. This type of mutation is most likely A. Nonsense mutation, B. Missense mutation, C. Silent mutation, D. Frameshift mutation, E. Transition mutation. Answer C. Silent mutation. A silent mutation does not result in a change to the amino acid sequence, often occurring at the third position of the codon due to the redundancy in the genetic code, tRNA wobble. This allows different codons to code for the same amino acid, leading to no change in the protein. In contrast, missense mutations lead to a change in the amino acid, and nonsense mutations result in early stop codons. 5. Which of the following mutations involves a purine being substituted with a pyrimidine, e.g. A to T or C to G? A. Transition mutation. B. Transversion mutation. C. Missense mutation. D. Frameshift mutation. E. Nonsense mutation. B. Transversion mutation. Explanation. A transversion mutation occurs when a purine is substituted with a pyrimidine, e.g. A to T or C to G, or vice versa. A transition mutation involves a purine being substituted for another purine, A to G, or a pyrimidine being substituted for another pyrimidine, C to T. Missense, frameshift, and nonsense mutations describe the effects of these base changes on the protein, not the specific type of substitution.